engine running.
Absolute genius.
Get this.
Welcome.
This is the show where we bring you
Science.
What that essentially means is
discoveries,
Advances.
Questions. Research.
technology. Unbelievable.
Without further ado,
This is The Naked Scientists. Hello, welcome to The Naked Scientists. This is the show where we bring you the biggest breakthroughs in science, technology and medicine with me, Chris Smith. And today, it's our final Titans of Science offering in this series with world-leading cancer expert Charlie Swanton. We'll hear how the latest developments in our understanding of cancer's mechanisms are shaping treatments and preventative measures.
From Cambridge University's Institute of Continuing Education, this is The Naked Scientists.
Charlie Swanton was born in Poole in Dorset on the 24th of February 1972. He attended St Paul's School before he continued his studies at University College London, and it was there that his interest in oncology, the study of cancer, began. He's a world-leading lung cancer expert, and he studies the management of metastatic cancer and also drug resistance and incurable cancers. He's won dozens of prestigious awards for his work. He's currently the Deputy Clinical Director at the Francis Crick Institute in London, the Clinical Lead in Cancer Research UK, and I'm delighted to say he's also our Titan this week. Nice to have you on the programme, Charlie.
Thank you very much indeed. It's lovely to be here.
We're going to talk about cancer, but let's go right back to basics first of all. When we say this word cancer, what does that word actually mean?
So cancer refers to essentially an uncontrolled proliferation in the human body that can breach the normal tissue barriers and in many cases can spread beyond those tissue barriers to other organs either through the blood or the lymph or direct invasion and result in what we call space-occupying lesions or metastases in other organs. The reason why patients die of this disease is due to a subtle metabolic and immune consequences that prevent the immune system from working effectively or drive abnormalities in the way in which we metabolize products from what we eat or how we digest those products through to just the consequences of the volume of disease on that underlying organ, which could be the liver, the lung, the brain, the kidney, etc., that ultimately lead to organ failure.
So if cells are disobeying the normal genetic instructions and they're just growing where they shouldn't, when they shouldn't, does that mean they can also disobey biochemical rules as well? They can basically make any of the things any other cell in the body can make and just start doing that whenever they want. So it's a sort of biochemical disease as much as a physical growth disease.
I think that's exactly right, yes. And indeed there was the effect that you've probably heard of called the Warburg effect, and cancer cells in general are much more dependent on glucose metabolism, glycolysis, the biochemical breakdown of glucose. And one of the sort of hallmarks of this disease is the uptake of glucose very rigorously by cancer cells. And we image that in the clinic using something called FDG PET, which is essentially a radio-labeled glucose tracer that's taken up by cancer cells more than surrounding cells. And so that helps us image where the cancer is in a patient's body. That is an example of the sort of biochemical consequences of a cancer cell.
But certain bits of my body, the healthy bits, are talking to other bits chemically. They're sending signals through nerves in some cases, but through the blood, for example, with hormones. So do cancers also have recourse to that? Can a cancer influence another organ via hormonal signalling, for example?
Well, that is a great question, and it's a question that we are thinking very deeply about actually at Cancer Research UK in our Cancer Grand Challenge scheme, which is an international grant programme. And here we're doing exactly that, which is trying to understand the sort of systems level changes that occur in patients with cancer. And we're setting questions to address some of those problems. I'll give you one example, which is cancer cachexia, which refers to weight loss, either of fat or of muscle, in patients with cancers like pancreas cancer or cancers of the upper gastrointestinal tract, so gastric cancer, for example, esophageal cancer, and even lung cancer. Actually, cachexia is quite common in. And patients lose weight, and it's unclear why or how this happens. And we've got increasing insights into this metabolic process. And as you say, this likely comes about through mediators, potentially hormonal mediators that either are released from the tumour or surrounding tissue in response to the burden of disease that suppresses appetite or makes a patient feel sick or nauseous and ultimately reduces their calorific intake and probably also potentially increases their metabolic rate, as it were, to degrade muscle and fat to presumably liberate the building blocks that are required for the cancer to grow and divide.
So the cancer is effectively turning itself into the primary thing and using the body almost parasitically. It's growing off the body.
Yeah, that's the way I see it in some cases. And I think it's quite a scary proposition. It's very important to say right from the outset, there's no reason behind any of this. This is purely a reflection of genetic diversity in the evolving cancer mass. That is the number of cells and the differences between them genetically. We call this genome instability that comes about because of the inability of cancer cells to repair their DNA effectively, or the fact that they have ways of replicating DNA that's less faithful than normal cells, so you get more mutations. And it's that genetic diversity between cancer cells that acts as a sort of the substrate upon which so-called natural selection acts. And so everything we see in cancers is a reflection of that genetic diversity and natural selection playing out.
What fraction of the population, if we just pick the human race, what fraction of us are destined to have this happen to us?
Well, I think in Western worlds, we'd estimate that somewhere between 1 in 3 and 1 in 2 of us will suffer from or die from cancer now. We've made great inroads into the management and prevention of cardiovascular events and cardiovascular disease. Our progress in cancer and neurodegenerative disease has been slower. I see cancer as sort of the next big challenge that we're tackling after what I would state as a sort of revolution in our understanding and management of cardiovascular disease. And the community is doing an extraordinarily good job, but we still have a long way to go to develop new therapies, new means of early detection, and most importantly, prevention, new ways of preventing cancer.
The government announced this week that they're formally launching bowel cancer screening for people over 50 across the UK. Many countries also have similar sorts of screening programmes. Their rationale for doing that isn't just that it's a good idea to pick cancers up early. They're saying it's becoming more common in this particular age group. So why, at a time when we can do more, detect more, treat more, and we know more about all these sorts of diseases and what causes them, why are we seeing some things like some cancers encroaching on age ranges where previously, historically, they were much rarer?
There are some potential reasons why this is that are intriguing and potentially actionable. One is Western diets and the change in Western diets over the last 60 or 70 years, in the post-war era particularly. And some might say this could be just a reflection of increased energy intake. Others might advocate the possibility of ultra-processed foods that may be contributing to this. And then there are other potential causes that have been mooted, including more antibiotic use, change in the microbiome, and even pollution, potentially microplastics and pollutants that we ingest as part of our diet. There is no clear answer right now what the underlying cause of this increased prevalence is. I'm suspicious that there is something very peculiar and specific to diet. Why do I say that? Well, if you look at South Korea versus Japan, and you look at the incidence of bowel cancer in those two populations, in the under 50s, it's increasing in South Korea, but not in Japan. Now, ethnically, fairly similar, but quite distinct in terms of diet. And I think that gives us a certain clue that potentially there is something about the sort of Western diet that is slightly more prevalent in South Korea than Japan that may give us a clue as to why this disease is increasing in frequency in the younger age groups.
What do you think that those risk factors that you outlined, diet among them, are doing? What's the mechanism by which this translates into a cancer, whether it's a lung cancer, a bowel cancer, a pancreatic cancer?
To answer that question, we need to know what the reason is. So what is the clearest proximal epidemiological risk factor? We don't know yet. My suspicion is the Western diet. So then the question is, why would that be? Well, there could be a number of reasons, including how the diet might alter the underlying microbiome. We know that some bacteria in the gut can themselves cause mutations or induce mutations in the mucosa of the gut. Those mutations themselves are implicated in the earliest steps of tumour initiation. But I actually think it's more complicated than that. Particularly work from Alan Balmain and colleagues at UCSF has shown recently that most carcinogens that cause cancer in man don't induce mutations in mouse models. So they must be causing cancer through other means, and one of those is potentially inflammation.
But cancer is a genetic disease, isn't it? If I look at a cancer, I can find the cell is shot full of genetic changes by the time a person's got a well-developed cancer. So at what stage then does the cell acquire these genetic changes that make it misbehave? Are you saying there's something upstream of all of that then that makes it misbehave first and then it mutates?
If you'd asked me 5 years ago, how does a cancer initiate, I'd have said we've pretty much solved this problem. It initiates through acquisition of mutations, as you alluded to, that lead to mutations in certain genes that provide that cell with a proliferative advantage. Now, there are some problems with that model, it turns out, not least of all the fact that recent deep sequencing studies of normal tissue, normal skin, normal lung, normal liver, have found cancer-causing mutations all over our bodies, including in our eyelids.
Even in young people?
Even in young people. But certainly as you get older, you acquire more of these mutations. So if you take the eyelid as an example, the Sanger Institute, they did this very elegant experiment where they took multiple biopsies of the eyelid and subjected them to very, very deep sequencing, and they found many mutations that we traditionally associated with cancer.
You're saying then that the body is riddled with the hallmarks of cancer genetically all the time, everywhere, but it's not manifesting everywhere, in even a fraction of people who've got those changes.
That's exactly what I'm saying. So one of my good friends and colleagues at University of Colorado, James DeGregory, estimates that in a 60-year-old person, they will have 100 billion cells in the body with mutations we would normally associate with cancer. And yet, as I said to you earlier, our lifetime risk of cancer is somewhere between 1 in 2 and 1 in 3. Now, to give you an idea, we have something like somewhere between 3 and 30 trillion cells in the human body. So at the cellular level, cancer almost never happens. And yet 100 billion cells in our body have mutations in that you normally associate with cancer. So what's going on? And that's the multi-million-dollar question. And I think what we've realized over the last 5 years particularly is that cancer initiation is a two-step process. Those mutations may be necessary for cancer initiation, but they're not sufficient. So on their own, they rarely induce cancer, very rarely induce cancer. Instead, what's required is you need a second step, which Isaac Berenblum referred to as a promoter step. And that promoter step can be induced by tissue inflammation, by chronic inflammation. So we've seen this, for example, with exposure to air pollution. So air pollution gets into the lungs, it gets taken up by white cells called macrophages, and those macrophages can't digest the air pollutants and churn out these inflammatory signals that signal, we think, to a stem cell pool in the lung with a cancer mutation to turn into a cancer.
So if we take asbestos exposure as an example then, this is a particle that gets deep into the lung, the body can't break it down, and it basically frustrates the immune system trying to get rid of something it can't get rid of. It churns out loads of these inflammatory signals, so you have chance damage genetically to some nearby lung cells, and you have all this inflammation. Put the two together, and then you've got a risk factor for getting a cancer.
That's exactly it. Mesothelioma, I think, is the most salient example of this process. Mesothelioma is a disease of the pleura around the lung. As you rightly said, the commonest risk factor for this is asbestos exposure. These small, fine asbestos fibers get deep into the lung. They get taken up by white blood cells and other cells, causing chronic inflammation, chronic release of inflammatory signals like interleukin-1 beta, the same inflammatory signal we found in response to air pollution in the lung tissue from the white cells I mentioned, the macrophages, that happen to act upon cells that just by chance have the wrong mutation in the wrong cell at the wrong time, exposed to the wrong inflammatory mediator. I liken it to a sort of Swiss cheese effect. It's lots of things going wrong simultaneously.
That does align with what we've seen from the use of drugs like aspirin, doesn't it? With people who have colon cancer, we know that if they have aspirin around the time that they're treated for that, their chance of it coming back is lower. Is that because it's suppressing inflammation? So if you've still got some of those cancer-promoting mutations lurking there, the chance of that 1-2 punch, that Swiss cheese model with the holes lining up, it just got a bit lower.
I think that's my assumption. I think we have to prove that formally, but that's certainly what we're thinking, yes.
So is the answer to cancer then we all just put ourselves on anti-inflammatories?
I wish it were that simple. I mean, I think for us, what these new findings over the last 5 years are revealing are opportunities to do that. The problem is we would need to find common inflammatory regulators where one could intervene and offer a drug to suppress them. And right now, we don't know what those are. We've got some very, very strong clues, but I think that's essentially what our lab is going to be turning its face towards working on for the next 10 years, to try to understand what those nodal regulators of tissue inflammation are that are allowing those holes in the Swiss cheese to appear, so to speak.
In the last 5-10 years, researchers in a number of labs, I spoke to one of them in Israel, for example, have been studying bacteria that appear to be associated with cancers, and particularly cancers that are wont to metastasize or spread. Is that just an effect because you've got an abnormal cell and it's grabbing some bacteria? Or are the bacteria really doing something? Because they were making a case for it may even cause resistance to chemotherapies of some kind, so that it's like the bacteria are helping the cancer cells.
Yeah, I think this is a very, very interesting area, and an area that is emerging quite rapidly, and the evidence is building very steadily to support just that. Let's start at the beginning. That one very clear bacterial cause of cancer is a very common cause, H. pylori in the stomach, that causes stomach gastric cancer. There we know very clearly from the work of Barry Marshall and others, Nobel Prize winner, that H. pylori directly causes gastric cancer. Now, over the last 5 years or so, it's emerged that another bacteria, what's called so-called PKS positive E. coli, I mentioned earlier, causes mutations in the gut that probably themselves help to initiate that first early event in cancer, help initiate those mutations in the cells that possibly allow other tissue inflammatory mediators to start driving that first cancer invasive step. Now, as you rightly say, we've reported over the last few years the association of the gut microbiome with differential response to therapy and presence of bacteria actually in tumour cells. What we're lacking, I think, right now is very clear functional evidence of how that process works. It's clear it's complicated. For instance, how does a gut microbe influence the peripheral response to immune therapy? How would it activate or repress, depending on the type of bacteria, the cellular response, the T cell response to immunotherapy in the periphery, i.e. in the bloodstream or in the lung, distant from the gut? Understanding that process is tricky, and it's taking time for that evidence to build. But it's building fast, and I think it's going to be a very hot area over the next 5 years.
Some people say that the microbiome is one of those hidden organs that we overlooked for far too long. There's 50 trillion bacteria in the average human gut, with a whole repertoire of molecules that they secrete into our circulation. I mean, the John Cryan in Ireland, for example, is showing that this is even involved in forming the blood-brain barrier in a developing baby inside a pregnant mother. So if you've got those sorts of signals, it doesn't seem beyond the realms of possibility that if you manipulate the microbiome, it could manipulate a cancer.
I think that's absolutely right. And I think what science has taught me in the last 20-odd years is you have to believe in the impossible, frankly. Stuff that's right now feels like science fiction tomorrow becomes science fact. Again and again this happens. And it goes back to what you mentioned earlier in our conversation. I didn't, 20 years ago think of cancer really as a systemic disease. That is, the tumour cells interacting with the body more broadly. The tumour cells interacting with the liver to affect metabolism, or breaking down muscle and fat to create building blocks for its growth, or even communicating with neuronal networks, for example. This is a disease that's truly systemic and interacting very broadly with multiple organs. And I think the same can be said for the microbiome in the body too.
Let's talk about how we actually manage it or treat it, because understanding the mechanism of a disease is absolutely crucial and fundamental to treating it, because it gives us insights into how we can basically put a spanner in the works, doesn't it? Yeah. So what's been the really big steps forward that we've made using our understanding of once the cancer snowball is rolling, where we can get in the way and stop it now?
That's a big question to answer. I mean, I think if we just, first of all, focus on the major breakthroughs in the last 2 decades, which is pretty much when I started practicing medicine, I'd say that the first is the advent in my field of targeted therapies. What does that mean? These are small molecules, pills which you take, or antibodies which are given into your veins. that interact as specifically as possible with aberrations in the cancer cell. They could be mutations in particular genes or resulting from overexpression of particular genes leading to protein overexpression on the cell surface that allows a cell to proliferate more. Example being HER2, common breast cancer oncogene that's targeted by the well-known antibody called Herceptin. Those, I think, represent very major breakthroughs, and I think the poster child of that would be imatinib, for example, in CML leukemia, which targets very specific kinases that are abnormal in the cancer cell but not in normal cells. That's targeted therapies. And moving on from targeted therapies, in the last 10 years, maybe 15 years, the biggest and most exciting breakthrough, without a doubt, I think, and most people would agree with this, would be the advent of immunotherapies, something that was sort of largely sidelined in the 90s when I was training. People had sort of given up hope of boosting the immune system to target cancer, and many funders have withdrawn funding from the field until Jim Allison and Honju published their work showing that there are breaks on the immune system that can be identified, namely CTLA-4 and PD-1, that you can target with antibodies and inhibit, which releases the brakes off the T cells, the immune cells, that allows the immune cells to recognize the cancer and kill it. And that was really, made very clear in trials led by colleagues, friends of mine like James Larkin at the Marsden, who showed very nicely in large studies in melanoma. Melanoma doctors now talk about curing patients with advanced melanoma, metastatic melanoma, even patients with melanoma that spread to the brain, with drugs that inhibit PD-1 and CTLA-4. And they work, as I said, by releasing the brakes off the immune system that allows the immune cells to recognize the tumor cells. This has been absolutely staggering. Absolutely staggering. I remember being in clinic with a mentor of ours back in 2005, 2004, Professor Martin Gore, a phenomenal medical oncologist, who at the end of a clinic on a Friday evening just said how sick and tired he was of treating melanoma, a disease that they'd made no progress in his entire career, over 40 years. And then within 18 months, the first trials of these immunotherapies were coming out through the Royal Marsden Hospital and other hospitals, showing these spectacular responses and patients being put into long-term remission with the disease. Almost overnight, the management of this disease changed. That's what gives us all hope working in this field, is that what seems impossible now, tomorrow is suddenly made possible. And I think it gives patients and doctors real hope for the future.
The obvious question from all of that is, if the immune system can do that, why doesn't it?
Well, I think it does. And the tumour finds ways of subverting it. First of all, one of the clear ways that the immune system fails to recognise a tumour cell is because the tumour cell downregulates the expression of surface proteins called HLA, human leukocyte antigen, also known as MHC, which essentially allow the body to distinguish between self and non-self. And due to the processes I mentioned earlier of genetic diversity and selection, you've got this sort of predator-prey relationship. The predator is the immune system and the prey is the tumour cell. Well, the tumour cell evolves to adapt and avoid the immune system by subverting those molecules on the cell surface that allow the immune system to recognise self from non-self. And that's one way that they can evade active predation by immune T cells.
Therefore, the solution being we just switch off that brake on the immune system, and that ought to deal with, all cancers? Or do you think that there are going to be some that will probably respond, but we're going to find some that are impossible nuts to crack?
It's another great question, actually. The number of tumour types that are responding to these drugs continues to increase. Even tumour types I would have guessed a priori would not be sensitive to these drugs are actually, particularly in the early disease setting. So even, for instance, estrogen receptor positive breast cancer, recent data has shown that patients are responsive to immune checkpoint blockade, even though these tumours generally have quite a low number of mutations. And I think that really raises some very important questions about what the immune system is actually recognising in these tumour cells. I don't think we've really bottomed that out yet. And whilst mutations are part of what they're recognising, there are other things too going on that we don't fully understand yet.
As our confidence increases with gene editing,
Yes.
Are we into a regime or a realm yet where we could see ourselves thinking, well, we know what's gone wrong with the cancer cell, we know where those cells are, and we can target them, we think, pretty selectively? Could we go in and gene edit them in situ, and that would take the cancer and any metastatic deposits down? Or is that just too risky?
By the time a patient presents with cancer, particularly late-stage cancer, the burden of disease can be quite large, and that is the number of cells can be very large indeed. Hundreds of millions of cells, billions of cells, and at death can be even larger than that. The challenge here is delivery. How does one deliver gene editing technology to that many cells? Because it only takes a few cells to escape for resistance to emerge, for example. You would have to target every single cell, and that's a real challenge.
But that's where the immune system comes in. So we have a sort of multi-pronged
potentially.
approach to this. So we hit them with the gene edit, hit them with the immune checkpoint inhibitors so the immune system can wade in. So we're kind of attacking it on multiple fronts.
Yeah, I think that's right. That's potentially exactly the case. That may work. I think this is early days. Gene editing is still in its relative infancy. It's hugely exciting, but delivery continues to be one of the major problems for gene therapy, gene editing technologies, and it has been really since I was in training, and it continues to be.
Are we slightly, though, going about this the wrong way because we're trying to slam the door after the horse is well and truly bolted? Should we not really be putting as much energy as possible into diagnosing people really, really early and finding ways to stop it there when it hasn't turned into billions and billions of cells? Because by then the hairs are running. We're not going to get this back.
Yeah, I mean, that's certainly the conclusion we've come to, that our work in TracerX and many other investigators across the world have shown very similar things, that there are so many evolutionary paths through which these tumours can grow and adapt through, that in late-stage disease, achieving cures is going to be very difficult indeed. I mean, obviously, I never say never, but I think that makes us think, well, how can we achieve the maximum good for the maximum number of patients in a short sort of 20-year career? And I think what we're doing now is turning the lab towards, particularly at early diagnosis, as you mentioned, but actually one step further back from that, which is, as I said to you earlier, thinking about routes into prevention. How can you attenuate tissue inflammatory pathways to prevent cancer from occurring in the first place? What are those inflammatory pathways? Can we block them specifically such that they will reduce your risk of cancer but still enable you, for instance, to fight infection? Because that's one of the key problems, that these inflammatory pathways are there for a reason, to protect us from microbes from outside, viruses, bacteria, etc. So we'd have to do this in a subtle way that can prevent the cancer from forming whilst maintaining our ability to fight bacteria and other viruses, for example.
You've mentioned during this interview in a few cases where you've completely changed or revised your opinion or changed your mind about what was happening with this disease. What would you really, really like the answer to? If I could wave a magic lab Petri dish in front of you right now with the answer lurking inside it, what would you really like to have the answer to? Perhaps to finish your career with, saying, well, I solved that one. What's the really big one?
Okay, there is one question I really want to answer, and that is the following. I mentioned to you about work from Isaac Berenblum and Alan Balmain of the two-step model to cancer initiation, which is you need those mutations and you need the inflammatory mediator. The next question, which I think is going to take us the best part of a decade to solve, and we may never solve it, others will, is how do those two collaborate? What is the inflammatory mediator doing to that stem cell with that cancer mutation? I think there is a switch. And if there is a switch that's switched on in cancer, I want to find ways of switching it off. Because if we could do that, we can get to the proximal very first step in how this all starts and stop it, potentially even in the latest stage of disease.
And when you walk out of the crick in the evening, what do you do to switch off?
I don't switch off. I carry on thinking about that problem.
Well, you've got a bottle of Shiraz on the desk over there. Was that not helping?
Oh, I'm not sure I should really talk about that, but yes.
It's a Christmas present by the look of it.
Yes, it certainly does help. It certainly does help to switch off, but your mind still wanders back to these problems. You know, you wake up at 2 a.m. thinking about it, and sometimes you have your best ideas when you've switched off, and you suddenly wake up and think, "Eureka! That's how you should do it." They don't happen quite as commonly as they used to, maybe 20 years ago. But this is such an exciting job. It's very hard not to think about this, even when you're relaxing. And even, we spend a lot of time as scientists talking to other scientists socially, because that's what we do. We love this job. It's got real purpose and real practical impact.
Stirring stuff and an inspiring note on which to end this series of Titans of Science. My great thanks to oncologist Charlie Swanton and also to the other leading figures who've spoken with us over the last couple of months. And if there's somebody who is a titan of science that you know and you'd like us to catch up with them, why not tell us all about them? It's chris@thenakedscientist.com. Now, in next week's programme, we're going to be asking whether it's possible to grow a building. It'll have you rooted to the radio, I promise. The Naked Scientist comes to you from the University of Cambridge's Institute of Continuing Education, and we're supported by Rolls-Royce. And talking of support, we also rely very heavily on support that you at home give us with your donations. And I want to say thank you very much indeed to those people who give us a regular donation and also people who've stepped up and made a one-off contribution. I've been writing to each and every one of you to say thank you. We are really grateful. And if you do like this programme and you'd like to show us your appreciation and help to keep the show on the road, please go to nakedscientist.com/donate. Every little helps. Thanks for listening, and until next time, goodbye.